When we talk about the endosymbiotic theory, the reason why this theory exists is it explains how we go from this prokaryotic cell that we're going to start out at the origin of life through several steps that are going to take several 100 million years to arrive at a eukaryotic cell. And the difference between these cells, if we look at them side by side, I know we just got these pretty simple drawings here, but prokaryotic cell has got all the same componentry. It can do all the same tricks for the most part that a eukaryotic cell can do. The difference is that the prokaryotic cell doesn't have anything on the inside in terms of membrane-bound organelles. All these things we see inside of the eukaryotic cell, things like the nucleus, the Golgi apparatus, the ER, mitochondria, chloroplasts, those are all membrane-bound organelles. And none of those are found in a prokaryotic cell. Those membrane-bound organelles are, first of all, what allows the cell to be much larger in a eukaryotic cell because of that compartmentalization, and it also allows for more complex biochemical pathways. So how do we go from this cell that doesn't have any organelles to this one that does? What are the steps that are involved? The endosymbiotic theory is something that was proposed by Lynn Margulis. She won the Nobel Prize for her work with this theory, and it's going to describe three of the most important organelles, their origins. And those are the nucleus, the mitochondria, and chloroplasts. And in general, we believe they happen in this order: nucleus first, then mitochondria, then chloroplasts. And when we're doing the very first step, which is the nucleus, you remember that we're going to begin with a prokaryotic cell. And because it is a prokaryotic cell, it doesn't have any membrane-bound organelles. Its DNA is within the cytoplasm. In this area, I'm going to use a dotted line to indicate this area, and it is called the nucleoid. I kind of messed up the spelling, but the nucleoid region. So it is not a nucleus. There is not a membrane enclosing the DNA, but there is an area where you find the DNA, and it's going to be really, really dense within the cytoplasm because we've got a lot of ribosomes and stuff in that region, and we call that the nucleoid region. Well, any cell can always benefit from having more membrane, because membrane allows for greater surface area to volume ratio. And so this prokaryotic cell, it's going to have invaginations of that outer membrane. So areas where the outer membrane is folded inwards and they're going towards the center of the cell. And the reason that these would have been beneficial is because now you're going to have all this extra surface area around the periphery of the cell that's going to allow you to bring more nutrition inside of the cell if you're absorbing nutrients. It'll allow you to get rid of waste more rapidly. And so it would have been beneficial to have greater surface area to volume ratio no matter what. Remember, evolution is not goal-oriented, but all these invaginations in this scenario are going towards this nucleoid region that has got our DNA inside of it. Well, the DNA, remember, that's like a physical object. It's something that you can touch and feel and hold and unfortunately break or damage. And so you can imagine that these invaginations would have been like little shock absorbers that are keeping that chromosome safe and preventing physical injury, and that would have been beneficial. And so with the endosymbiotic theory, we're going to say that these invaginations of the outer membrane are going to be helpful for that cell. They're going to protect the nucleus and also regulate what can go towards the DNA. And so if they simply break off, so imagine that this little part of the outer membrane just kind of pinches off from the outside, you would end up with that outer membrane still intact. Your DNA is right here, but instead of there just being like an area of the cytoplasm where you find the DNA, we've got this membrane that pinched off, and so it's going to form a second membrane on the inside that's surrounding the DNA. And this structure that contains the DNA is now our nucleus. So we go from a nucleoid region to a nucleus, and once you get that nucleus, this would technically have been a eukaryotic cell. So eukaryotic cells can have all sorts of tricks they can do, and we would have technically had this eukaryotic cell as soon as we get the nucleus, but we want to get these other things too. We want to get things like a mitochondria and a chloroplast. So for the mitochondria, remember that this is a prokaryotic cell, and at the origin of life, all cells would have been fermenters. They would have only been able to do fermentation. They wouldn't have been able to do the full suite of aerobic respiration. And so if you've watched the fermentation video and you've seen this kind of stuff, these are the only steps they can do. And remember that with fermentation, it's inefficient, it's wasteful, you only get 2 ATP for every glucose. So we're going to imagine a world where we've got this cell that we just talked about. It has a nucleus, And so here's our eukaryotic cell, and it has its nucleus in there, okay? But it's still going through respiration, which again is super inefficient. Well, somewhere else in this world, there's another cell hanging out, but this cell has been able to evolve aerobic respiration. And so that means this one is doing respiration, but it's doing it aerobically or with oxygen. So instead of getting just 2 ATP, like this guy, this one's getting 38 ATP with the same amount of glucose. So it's like, imagine you go to work and you're earning $7 an hour, and you find out the guy with the same job as you, showing up with the same uniform, is somehow getting paid like, what would that be, like $50 an hour. And you'd be like, what is this guy doing? I want to be able to do those things. And so this cell engulfs this cell. And again, it's not because this cell somehow knows what this one's doing, and it's, again, evolution is not goal-oriented, but this cell is going to engulf the other one, and that would have happened just because if this is a heterotrophic cell, that's how heterotrophic unicellular organisms make a living, as they go around and engulfing other cells. So it's going to engulf this guy. And so this one would be our host, okay? And this is going to be our aerobic cell. And so it's going to engulf this guy, okay? And when it engulfs it, every time that you import something into a cell with endocytosis or penocytosis or just bringing in another cell, phagocytosis, this membrane of the host that is surrounding the thing that we're engulfing, that membrane is going to pinch off and engulf this thing to the interior. So after the phagocytic event is done, you've still got your host. Your host has still got its nucleus, but it's just engulfed this cell, and that cell is engulfed by a second membrane from the host. So this membrane and this membrane are the same. This is host membrane, this is host membrane. But then this membrane to the interior is the same as this membrane over here that was originally on that aerobic organism. Well, in this scenario, we're going to engulf this aerobic organism, but this aerobic organism is going to stay alive. It's going to continue to perform aerobic respiration with the sugars that are floating around the cytoplasm of this cell. And it would have just started releasing ATP. ATP would have just been leaching out into the cytoplasm of this cell, much more ATP, produced much more efficiently than this cell ever could. And so rather than breaking it down and eating it, it's just going to keep it alive. And so we've got that host cell. It has its nucleus, and this cell that has now become a part of this cell is going to eventually become the mitochondria. Now that that inner membrane doesn't really need to have the same shape, it's being protected by this outer membrane, that inner membrane could invaginate and form a whole bunch more surface area, because when we talk about aerobic respiration, we talk about how this membrane to the inside of the mitochondria is really valuable. And so you would have gotten this new organelle, which is the mitochondria. And so with this scenario, your host would have been a fermenter. The thing getting engulfed was going through aerobic respiration, and it's retained in the host and then eventually becomes its own cell. And the reasons why we think this is how it happened is because mitochondria is surrounded by two membranes, not one, so that it lets us know that something was engulfed. And secondly, if this one was doing aerobic respiration, the host was doing aerobic respiration, it wouldn't have really gained anything by absorbing this other cell or engulfing this other cell. And so it must have been that this was the fermenter, very inefficient, and the reason why it retained this cell and eventually went on to become the mitochondria is because this one was so much more efficient than the host ever was. Now we got chloroplasts. It appears that mitochondria arose once. This whole event happened one time, and so we have one origin for mitochondria. Chloroplasts, on the other hand, have evolved multiple times. And so a lot of times when we're talking about these, we actually say plastids. And so plastids means that it's any number of phagocytic events that are going to lead to the origin of a plastid. And so the way that you get photosynthetic organelles in like green algae is different from how it would have happened in like brown and golden algae. And so we just say plastids to describe all of them. When we're talking about chloroplasts, we're typically talking about green algae. But the way this would occur is you've got your host, and in this case, it's your host has already got a nucleus, and your host has already got a mitochondria with that differentiated inner membrane. And it is a heterotroph, right? And so this cell needs to engulf other material in order to keep itself alive. Well, this guy is going to encounter a new kind of cell, and this one is called the cyanobacterium. A cyanobacterium. And cyanobacterium were the very first bacteria. So notice bacterium. This is a bacterial cell. It's a very, very small, simple cell. It would have been like this one. No organelles. But cyanobacteria were the first cells on the planet, the first bacteria to start doing photosynthesis. And so photosynthesis, that allows you to become an autotroph. You can produce your food with sunlight. You don't have to go engulfing other cells. You can see how that's really advantageous. So if you are this cell and you encounter this cyanobacterium, you would have eaten it, because remember, these are bacteria. This is a eukaryotic cell. So this eukaryote would have been much larger than this prokaryote. And so here's our nucleus, here's our mitochondria that we talked about before, and it's going to engulf that cyanobacterium. And just like we saw over here, every time you have a phagocytic event, you're going to add a membrane onto the thing that becomes engulfed. And so our eukaryotic cell is now going to have that cyanobacterium inside of it, but the cyanobacterium is going to be absorbed by another membrane. This membrane is from the host, and we've got our nucleus, and we've also got the double membrane-bound organelle of the mitochondria that's over here, and so it would have absorbed this cyanobacterium. Well, the cyanobacterium could have been producing sugars every time that was exposed to sunlight, and then those sugars, those carbohydrates, would have leached out into the cytoplasm the same way that this ATP would have been leaching out of the cytoplasm over here for the mitochondria, and that would have been beneficial. And so this host could have benefited by not engulfing or by not eating, dissolving, killing this cyanobacterium, and instead allowing it to survive within the cytoplasm of the cell. And so we would go from this just cyanobacterium that was engulfed to the more differentiated chloroplast that we see today, that has got the stroma inside, and it's got the thylakoid membranes, and it's got, again, a differentiated inner membrane. And so with chloroplasts, they look more like straight, and then wiggle, and straight, and wiggle, and straight, and wiggle. And then we have our nucleus over here. So notice that we get the nucleus from the original host cell, just an invaginated membrane, and that's why the nucleus is eventually going to evolve. But then over here for mitochondria, we've got two membranes surrounding that organelle because of this phagocytic event of an aerobic respiring cell. And then for the chloroplast, we have this phagocytic event of a cyanobacterium. And so once you have both of those things, you would have basically what we'd call like a plant cell, for example, which would have been like a green algal cell or euglena, that is going to be this kind of self-reliant cell that can make its own sugars and then produce ATP with those sugars. All of that together is the endosymbiotic theory, which was originally proposed by Lynn Margulis.